Epigenetics, which is gaining quite a bit of traction right now, is actually looking at how certain processes change the histones on DNA. Oops, I'll go back. Histones on the DNA, which then allow for changes to occur in the phenotype or the physical characteristics of that individual without any changes in the DNA. There was a study done at Johns Hopkins University looking at the agouti mouse. In this particular experiment, the researchers took all of the methyl groups out of the diet. All of the B-complex vitamins that could work through the folic acid pathway to produce methyl groups. Choline they took out of the diet. And when they did that, the brown mouse hair coat turned to yellow. So this was one way that the environment can influence the phenotypic expression of a particular animal based on what might be going on in that environment. For example, there might be some drought or devastation, or they have to find another food source. So this is, I believe, one way that we can adapt to certain environmental changes that are going on in our life. And in doing so, we don't have to change the DNA. So it's a quick modification of our phenotype in response to something that's going on in our environment. Functional genomics, on the other hand, looks at a carefully selected group of genetic variances in a person's genome. And we do that through a blood sample. There are many companies on the internet today that are doing genomic testing. They typically use a saliva swab to capture DNA through your white blood cells. And in doing so, they pick up these modifications. The companies that are typically like 23andMe, how many people have heard of 23andMe? Yeah, 23andMe is a, this is interesting, the owner, co-owner of 23andMe is actually the wife of a Google executive. So they have a lot of money behind them in promoting what they're trying to do. But you have to be very, very careful, because I call this informational genetics. Because all they're doing is listing the probabilities of whether you're going to get a disease process or not. So they may say, when you get this genomic result back, it may say 1 out of 200 you're going to have prostate cancer. Or 1 out of 300 you're going to have breast cancer. Well, that doesn't tell you much, because it doesn't tell you what you need to do. It just creates a lot of anxiety in people to make sure that they have to go to the doctor more often. The genome evaluations that I do, on the other hand, are very different. The genes that I identify are modifiable. Genes are modifiable. You can change that genetic expression either by a lifestyle choice, or a change in your diet, or you can use a nutritional supplement to bypass a particular SNP, or single nucleotide polymorphism. So these genetic expressions can be modified. And today we have biomarkers to make sure that when we do a nutrigenomic intervention, we can actually see if there's any change. One of the advantages here is that we get to see the potential risk of a disease waiting to happen in a person's genome, and then modify that expression so that we push back the chronic disease incidence to maybe the 6th, 7th, or 8th decade of that person's life, which gives that individual a tremendous advantage, so that they're not in a chronic disease state 30, 40, 50 years before they pass away. There are key points about the human disease process that I want to relate to you. All human diseases occur from an interaction between genes that are predisposed towards a particular disease process, their environment, nutrition, and lifestyle factors. And in these slight variations that are associated with DNA are called SNPs, single nucleotide polymorphisms, meaning that the nucleotide has shifted. And I'll give you an example of this. Because when you look at these single nucleotide polymorphisms, there is the potential expression of a disease. And it depends on whether that disease resides, or that SNP resides on either one chromosome or two chromosomes. There are more than 30,000 genes, yet 2 million SNPs. The SNPs actually convey our uniqueness. So they're not bad. They're actually really helpful because it gives us the genetic variation that we see in this room today. This is an example of what happens when you see a normal sequence of DNA. And remember that there are 4 letters that are associated with nucleotides, A, C, T, and G. And they bind with each other in a specific way. The G, guanine, binds with cytosine. Thymidine binds with adenosine. When you have a single nucleotide polymorphism, that is a switch in your DNA, just brought about by aging, just brought about by being on this earth. Because when the DNA replicates, it makes mistakes. And if it substitutes a TA for a GC, then things can happen downstream. This is an overview of what happens when the DNA sequence can vary. For those of you who have not had a genetics course recently, the 3 nucleotides together are called a codon. This codon then expresses, is basically the language for a particular amino acid. In this case, it's alanine. In the next case, it's arginine. So when this individual has a normal expression of these codons, creating these amino acids, it forms a protein. The protein then has biological function in the body. It can be a skeletal protein, it can be a receptor, membrane protein, or it can be an enzyme. In the second case, where there is a substitution here, the adenosine is, a guanine is substituted for the adenosine. It still makes the amino acid alanine, which then forms the normal protein. So there's no big change in that protein expression. Yet in the third example, in this case, the adenosine is substituted for this guanine on this second codon. Instead of making arginine, it makes lysine. And in doing so, it changes the conformation of that protein. It changes the shape of that protein. So if it's an enzyme, oops, you could have some reduction in that enzymatic activity. If it's a part of a skeletal protein, like collagen, it could damage the ability of that collagen to be made. Or if it's a receptor protein on a membrane, then it may not be able to recognize hormones and communicate with the outside world. So the whole concept here is to better understand what's going on in the genome, to understand how proteins are being synthesized, and cell function can relate back to that. There's a big push right now to work backwards, looking at what cell function is, how it relates to a particular protein or proteomics, and then trying to go back into the nucleus to figure out what's going on with that particular DNA.